Hello, we'll continue our series of videos on reporting microscope metadata with Micro-Meta App and MethodsJ2. This section discusses mostly MethodsJ2, a software tool to capture metadata and generate comprehensive microscopy methods text. And in this video, we'll give a quick overview of how MethodsJ2 works and what are its data inputs, including the Micro-Meta App hardware specifications file. So the goal of MethodsJ2 is to generate methods section text. And so where does it get that information for text generation? Well, first, as you might imagine, information contained in the image itself, image metadata, is used and is sourced using bioformats. And then MethodsJ2 also requires a Micro-Meta App hardware specifications file, which contains detailed information on components of the microscopy system. Micro-Meta App was described in a previous video by Caterina Strambio de Castilla, but I'll give you a quick reminder here. Micro-Meta App allows users such as core facility staff, imaging scientists, and so forth to build a microscope in silico, generating a database of all the components on a microscope. This database is generated using a standardized microscope metadata model known as the NBO model. And the output of this is a machine-readable microscope configuration file, and more specifically a JSON file, a .json file, which is then used to essentially store all the information that's put in. This JSON file therefore contains all the information about the components and specs of the microscope itself. So how does MethodsJ2 work? Well, as we mentioned, we need an image, ideally an image that contains metadata from the microscope itself, as well as a Micro-Meta App hardware specifications file, a microscope hardware specifications file. These files are taken in by MethodsJ2, which is a Python script that runs in Fiji. And so Fiji, the open source image analysis software. MethodsJ2 then collects information from the metadata files, the image metadata and the Micro-Meta App hardware metadata files, and then prompts the user to validate or fill in information about the imaging experiment through a series of dialog boxes. This information is then used to generate a methods section text, which can then be copy-pasted into a manuscript. So that's it for the quick overview of MethodsJ2. In the next video, we'll review the materials needed for the demo, and then we'll move along with the hands-on demo itself.